agenda is as follows. Doors open, national anthem of the Republic of Colombia, words by the Consul General of Colombia in Chicago. Welcome and introduction to the Colombian College of Neuroscience, Colne, and Neurodiaspora Colombia Initiative. Recognition to the Brain Initiative and Dr. John Nye by Colne and Neurodiaspora Colombia. Words by Dr. John Nye. Awards to the best papers by a Neurodiaspora member in 2024. And recognition to Dr. Francisco Lopera. Then a panel on career development for Colombian neuroscientists abroad. Then some wine and cheese and concluding remarks. Bueno, entonces damos inicio al himno.
Comprende las palabras del
A continuación, unas palabras de bienvenida del doctor Diego Angulo Marínez, cónsul general de Colombia en Chicago.
Buenas noches.
Buenas
Un
noches.
saludo especial a todos los organizadores de este encuentro de neurociencias, neurodiáspora científica de Colombia en los Estados Unidos. Es para nosotros en el Consulado General de Colombia en Chicago un honor y un placer compartir con ustedes por muchas razones significativas. La ciencia es el motor por excelencia del desarrollo científico de la humanidad. Colombia es un país que se encuentra a la vanguardia de encontrar fórmulas para poder crecer en un ambiente en el cual los nuevos desafíos de estos tiempos lo ameritan con rigor. Que ustedes hagan parte de esa generación que, como una playa de figuras prominentes, se encuentra a lo largo y ancho de la geografía de los Estados Unidos de Norteamérica, y que con ese ímpetu que hacen ciencia puedan ser nuestros embajadores para concretar todo lo necesario en la búsqueda del conocimiento, es algo que nos enaltece y que nos lleva a decirles que los instamos de una manera significativa a que sigan contribuyendo al país de la manera en la que lo vienen haciendo. Sabemos que hacer ciencia es algo que no está bien ponderado, que muchas veces no es bien remunerado, que no se genera el reconocimiento y el esfuerzo que ustedes merecen debido a las largas horas de investigación, los años de trabajo concatenado, la paciencia con la que realizan las actividades y el método por el cual han escogido esta actividad, que requiere de sacrificios personales, familiares, de esfuerzos económicos y de una tenacidad y constancia que los hace merecedores de un digno reconocimiento por parte de este cuerpo consular. En nombre del gobierno colombiano, nos sentimos orgullosos de la labor que vienen realizando. Creemos que ustedes son unos portaestandartes de esa misión. Y como dice el método científico que ustedes enaltecen, no solamente es conectar esas células del sistema nervioso y conectar todo lo relacionado con la médula espinal y el cerebro, sino también hacer un trabajo transdisciplinario que nos pueda albergar la certeza de que en el trabajo científico que vienen haciendo van a encontrarse con diversos saberes sociales, entendiendo que no hay verdades absolutas, entendiendo que la ciencia es un método tal vez infinito de conocimiento, que como diría Francisco José de Caldas, nadie es más ignorante hasta que entiende cuánto más sabe y cuánto menos conoce. Y de esa manera nosotros creemos que el aporte colombiano va a ser muy importante. Un aporte que se refleja en el gran aumento de connacionales que llegan a este país. Y además que con el avance de ustedes y la llegada de exponentes científicos e investigadores a universidades como las Ivy League y otras universidades de renombre en los Estados Unidos de Norteamérica, puede hacer de que Colombia encuentre un sitial muy importante en el concierto de saberes, en el concierto de lo que tiene que ver con la tecnología, unida con lo que hoy está en boga, como la inteligencia artificial, como la generación de conocimiento a través de otros métodos que nos permitan encontrar una verdad que puede ser relativa, pero que a la vez puede ser una verdad constante y una verdad que nos refleje no solamente esa introspección con lo que es importante para entender, sino con lo que es importante para descubrir y para proyectar. Esa es su labor, eso es lo que ustedes vienen haciendo a lo largo del tiempo. Ese es el compromiso social, es el esfuerzo por el cual han venido trabajando durante años. Y nosotros en este consulado estamos prestos a servirles, prestos a abrir las puertas de este consulado cuantas veces sea necesario, a trabajar con ustedes de una manera concatenada, en una colaboración armoniosa, en una coordinación, en una amplificación de nuestros conocimientos y en un servilismo importante, en un servilismo constructivo, en la construcción y en la proposición de la ciencia más allá de lo que conocemos, pero además de lo que más allá de lo que conocemos, en la proyección de lo que debe ser una humanidad que respete la vida, una humanidad que entienda que la ciencia no tiene que estar al servicio de las cosas, de los poderes, sino que debe estar en servicio del ser humano, de que el ser humano hoy que encuentra la tecnología y que encuentra la inteligencia artificial como algo que lo desborda, no los vea como algo que nos vaya a autodestruir, sino como aliados para la búsqueda de una humanidad mucho mejor, que supere su tiempo, que al superar su tiempo crea que sea posible avanzar en lo que hoy queremos que sea el quid del asunto, salvar la vida, salvar el planeta, construir una humanidad que sea mucho mejor. Bienvenidos al Consulado General de Colombia en Chicago. Muchas gracias.
Bueno, por parte del Colegio Colombiano de Neurociencias y del Grupo de Neurodiáspora Colombia, nos place mucho presentarle al doctor Diego Angulo Marínez este certificado de gratitud por todo su apoyo con este evento. Muchas gracias.
Unas cortas palabras. Este es un reconocimiento inmerecido y gracias por su generosidad. Yo quiero hacerle extensivo esto a todos los miembros del equipo consular que han hecho posible que esto se haga realidad. Por aquí hay varios, Silvia Juliana Uribe, Diego Luis Mosquera, tal vez veo a nuestra multiplicadora Gladys Zárate, a Vanessa de Villafranco, a Julián Méndez Pinilla, a Ana Camila, bueno, todos los que hacen parte de Lina Lara, los que hacen parte del equipo consular y que con su diligencia han venido a trabajar para que esto se haga realidad. Y yo lo que soy aquí es un vocero, un facilitador. Gracias.
Please. All right, so we're officially switching to English now. All right, so now we will give the floor to Dr. Pablo Reyes, who will provide the welcome and introduction to the Colombian College of Neuroscience and Neurodiaspora at Columbia.
Okay, thank you and welcome to this night. I'm going to read, sorry, bad memory. And thank you and the council, thank you for receiving us and welcome to this night. This is about an honor and privilege to be here today, representing Colombian College of Neuroscientists and this important event. I want to start by recognizing the effort, passion, and commitment of so many Colombians who from abroad work tirelessly to continue contributing to the advancement of neuroscience. Your achievements not only enrich the scientific field globally, but also elevate the name of our country, showcasing to the world the talent and dedication that define Colombian neuroscientists. To those who have chosen to continue their careers outside of Colombia, I want to express my deepest and most heartfelt gratitude. I know that deciding to settle it in another country is not easy. It requires tremendous courage, sacrifice, and perseverance. Many of you face it and overcome personal, family, and professional challenges in pursuit of better opportunities to advance the science. Those sacrifices, often involving the hardship of being far from your loved ones, have enabled your contribution to become an integral part of global scientific progress. Your determination to push forward despite the challenges is a source of inspiration for future generations of Colombian neuroscientists. Furthermore, you have become ambassadors of Colombia within global academic and scientific community. Through your research, collaboration, and mentorships, you continue to build bridges between institutions, countries, and continents, strengthening a network of knowledge that benefits all. Your success abroad not only represent individual accomplishments, but also open doors for other fellow Colombians seeking opportunities in science. Thank you for you carrying the pride of our country with you and demonstrating that no matter the distance, your heart remains connected to Colombia and to its development. I would also like to thank those who have chosen to return to our country. Upon your return, you bring with you a valuable knowledge, experiences, and perspective that really enrich our academic and scientific environment. You represent the bridge between what has been learned abroad and the opportunities for growth and development within Colombia. Your decision to come back is a testament to your deep love for our homeland and your commitment to its progress. In this context of collaboration growth, both from within and from abroad, neuroscience in Colombia continues to advance. Together, we are building a strong network, united by a common goal, to improve people's lives through understanding the brain and its complexities. No matter where are in the world, we are all part of this mission. The Neurodiaspora Culture Organization cordially invites all those interested in the field of neuroscience to join our network. As a member, you will have an access to a knowledge sharing platform collaboration and opportunities, and vibrant community of professionals and students. We invite to participate in our next Congress in Bogota to be held in April the next year. Really, thank you.
So thank you so much, everyone, for being here. Do you all hear me in the back?
Yeah.
Okay, thank you. So we are so proud and so happy to have John with us. John is the director of the Brain Initiative at NIH, where I serve as program officer and in many, many roles. And I had the pleasure to work with him for almost 2 years as lead for systems neuroscience. And under his leadership, we have been really, really advancing what we can do in the Brain Initiative. So I'm beyond excited that in this small event of a country in the middle of nowhere, and he is really deciding to come to be with us. So thank you so much. And we want to present this with the help of
Okay.
I don't know how you say console, the console. I was like,
Okay.
I don't know how you say that in Spanish, in English. So this is a diploma recognizing the
Yeah.
leadership and the labor of John Knight. Thank you so much.
Thank you so much.
Dr. John.
This is in the name of the Colombian government, Polne, and the Colombian Neurodiaspora.
Thank you so
On
much.
behalf of the Colombia people,
Thank you.
congratulations.
Oh my God. Thank you.
Congratulations.
And this,
Oh my goodness.
this also, yeah.
Great. Yep.
Oh,
Well, we can go this way.
great.
Thank you so much. Thank you. Wonderful.
Thank you so much, everyone. And now we're gonna give the floor to John.
Thank you all for being here. It's really quite an honor. Delighted, and I've enjoyed so much working with Maurizio as a colleague on the Brain Initiative. Do we have the slides up? Do you control? Do I control?
You say me next. And...
Okay. We'll try. We'll try. So I'm very excited to tell you about the NIH Brain Initiative, and we are now celebrating a decade of existence. The initiative was launched in 2014, so a decade of innovation. So if you can go to the next slide. Okay, great. So the mission of the NIH Brain Initiative is to revolutionize our understanding of the human brain by accelerating the development and application of new and innovative technologies. And as I'm sure you will appreciate, the human brain is composed of 86 billion neurons, maybe twice as many non-neuronal cells, making trillions, if not hundreds of trillions of connections. And we really don't yet have the tools to understand how all this works to give us normal behaviors. And certainly we don't know enough about what's going wrong in neurological and neuropsychiatric disorders. So again, we are celebrating 10 years of innovation. At the NIH, the Brain Initiative is a trans-NIH endeavor involving 10 of the 27 institutes and centers at the NIH. So next slide, please. So to achieve this goal of revolutionizing the understanding of the human brain, we are working toward leveraging emerging technologies to enable new discoveries about neural circuit function, to use these discoveries as a foundation for new therapies for curing human brain disorders, and very importantly, to disseminate and democratize these resources across the world for the benefit of all. So if you can go to the next, please. So our program is organized in 8 overlapping areas, and Maurizio is helping us lead one of these areas: brain cell and circuit technologies, better ways of mapping, monitoring, and modulating neural circuits, part of our neural recording and modulation portfolio, neuroimaging across scales from the mesoscale to the global scale down to the microscopic level, systems neuroscience and human neuroscience, which Maurizio is so deeply involved in. And then cross-cutting data science and informatics. We're generating a lot of data that need to be managed. Training, inclusion, and equity. We want to make sure we're supporting a diverse workforce, but also very importantly, supporting the mentorship and the cultivation of the next generation of leaders. Neuroethics is very important. The ethical, legal, societal impacts of what we do, especially as we get into human neuroscience, is becoming more and more important. And then again, the dissemination and commercialization of the technologies and resources that we're helping to develop. So the next, please, you go one more. And then one more, thank you. Okay. And again, we're just celebrating a decade of innovation. I wrote this little piece in this latest issue of Neuron, looking back at what we've accomplished, but really looking forward to what more we can do in the next 10 years. So if you're interested, you can find it online. It's open access. So next slide, please. So one thing we've been doing at the Brain Initiative is we've invested very heavily in developing large resources that can be used by the entire neuroscience community. This involves a lot of coordination and team science. We're generating a parts list, cell atlases of whole mammalian brains. The groups we have funded have just published a complete atlas of the entire mouse brain and draft cell atlases of the non-human primate and human brains, great resources for understanding the cells that are involved in neural circuits. We are working on this Brain Connects network to create wiring diagrams of whole mammalian brains. And right now we're just developing the technologies that are needed to scale up this massive endeavor. The repair tools, this refers to our armamentarium for precision brain cell access, basically building tools that allow experimenters to more precisely target cell types in these circuits and to modulate their function and to record what they're doing, ultimately with the goal of understanding how neural circuits give rise to behavior, but also how we can modulate them eventually for the treatment of human circuit disorders, hopefully one day with precision gene therapies. And then finally, this brain behavior quantification and synchronization program aims to develop better tools for monitoring behaviors in both humans and non-human models, to synchronize this with neurophysiological data to get a better idea of the cause and effect relationships between neural circuit activity and behavior, with the ultimate goal of developing closed loop therapies for behavioral disorders. So I'll just go through these projects in turn very quickly to give you a flavor of the kinds of projects we are supporting, again, for the benefit of the entire community. So next slide, please. So we've been investing very heavily since the beginning of the initiative in this brain cell census project. Recently closed out the Brain Initiative Cell Census Network, which led to the publication of some major groups of papers in Nature and in Science sister journals. Initially in October 2021, we provided a detailed multimodal characterization using transcriptomics, epigenomics, physiology, anatomy, and so forth in just a tiny part of the brains of non-human primates, humans, as well as mice in the primary motor cortex. This kind of gave us the know-how for conducting these projects, which led to the whole mouse brain atlas that I referred to before as 10 articles in December of 2023, 9 of which were funded by the Brain Initiative, as well as the draft human and non-human primate brain cell atlases published in October of 2023. And this is really just marks the beginning to provide the resources for people to use to functionalize these discoveries. Now the big challenge is through the Brain Initiative Cell Atlas Network, which is ongoing today, which is focusing on creating a complete atlas of all the cell types in the human brain. And right now the challenge is to move from data into a knowledge base. And already these data are being used to address specific disorders of the nervous system. So next, please. So in the case of addiction, next, please, 2 programs are being promoted by the National Institute on Drug Abuse to use data about brain cell types and the connections to better understand the mechanisms underlying addiction. And then in this case, in the Seattle Alzheimer's Disease Brain Cell Atlas Project, this is a large consortium of investigators in the Seattle area, now funded by the National Institute on Aging, that is using the tools and resources developed in the Brain Initiative Cell Census Network to look at the cellular changes that occur over the progression of Alzheimer's disease from early stage to late stage. And they found some very surprising findings about the cell types that are being affected at different stages, and interestingly, specific interneuron subtypes that are being affected very early in the disease process. And they'll go back a moment if you would. And now this paper will be coming out in, I think, next month. And the reason why this is important is because, as I'm sure you know, people have been looking at the genetic drivers of this disease. And of course, Dr. Francisco Lopera spent his lifetime first identifying in the large cohort of families in Colombia the PS1 mutations, and then later, and unfortunately he didn't himself see this to completion, has found genetic modifiers that might protect certain individuals from this devastating disease. And I think what these cell atlases will give us, the next generation of researchers, is now to look at where these genes are acting in the different cell types and hopefully complete his work that he dedicated his entire life to. So we're very enthusiastic that we can provide tools that will allow researchers who have built on many, many decades of research to actually find the root causes of these diseases and therefore to find not just treatments, but cures and prevention. So we're very excited about this, and I hope you all will be able to use this information as you kind of pursue some of these endeavors. So next slide. We've also just started this Brain Connects program, now in our second year, a large consortium of researchers across many countries that is developing the technologies that we will need to create whole mammalian brain cell wiring diagrams. Next, please. And we're pursuing this using three basic technologies. One is electron microscopy, serial electron microscopy. The other is barcoding sequencing tools to give us higher throughput and cellular specificity. And then a new optical methods to be able to map out on large brains, human and non-human primate brains, to project large, long distance projections of these different connections. So we're very excited about that. We are hoping over the first 5 years of this project, we'll identify the technologies to do it, and then in the following 5 years, actually to be able to create whole brain connectomes. Now, this is a big endeavor that will require technologies not yet in hand, but we're very excited that just this last week, the Flywire Consortium published the first full connectome of an adult Drosophila brain. So this is the first complete connectome of a complex brain, published as 9 papers in Nature. And what we're showing here are these beautiful renderings of this connectome, but highlighting these different specific sensory motor pathways. And now researchers are able to go through this and make predictions about which cell types and which connections underlie specific behaviors and test these both experimentally as well as computationally. So you can just go through the next 3 clicks. Very, very exciting. It's gotten a lot of attention, and it really does mark a milestone in the connectomics era. And you can keep going through. They identified close to 140,000 neurons of over 4,000 types, making over 50 million synapses and axons that encompass, I think, 150 meters worth of distance, which is really quite incredible. From a biological standpoint, the Drosophila has been for many decades a beautiful model for understanding the genetic and neural circuit basis of behavior. And we'll learn a lot about the fundamental principles of neural circuits, how they underlie behavior. But from a technical standpoint, many of the techniques used, especially in the artificial intelligence realm, to segment the data are now being scaled up and applied to the mammalian connectome. So this really will be the forerunner to the larger connectome projects in mammals. And in fact, many of the folks involved in this tremendous study are also involved in our Brain Connects project. So next, please. So again, this project, you can click one more, really embodies what the Brain Initiative is all about. The power of team science, the ability to create these resources that no one lab, no matter how talented or large, could do by themselves. The power of citizen science. The segmentation algorithms could only get the researchers so far in assembling these maps from 7,000 serial sections with 21 million images, and they still needed to be proofread by humans. And before they applied this algorithm that they used, the estimate was it would have taken 4,000 person years to proofread, so that's kind of ridiculous. The newest algorithm they applied to this brought the number down substantially, but still 33 person years. And the way they approached this is through crowdsourcing. Hundreds of individuals went in and proofread it, and they also involved citizen science, people that weren't scientists per se, but had an interest in doing this. So this is very exciting, and I think these kinds of principles will be applied to these larger efforts, both as a way of accomplishing these projects not just sooner, but also engaging the public and showing them firsthand how important science is, and very importantly, how much fun it can be to make people more interested in what we're doing. And then finally, the power of open science. This project was 10 years in the making, but starting about 5 years ago, long before this was published, they were releasing the data publicly. Anybody could access it, and actually it led to the publication of about 50 papers using these data before the creators of this map actually published their own papers. So we really are supporting the power of open science here. So next slide. And you can click through 3 times. And then the next large project we're doing is developing this toolkit of access reagents, right now still focusing on adeno-associated virus, to develop better ways of targeting specific cell types, to develop better payloads, whether they're going to be sensors or switches or gene editors, and then making them available to the general public. And right now on the AdGene website, AdGene now is distributing many of our reagents to anybody who wants them. And the last I checked, there were over 2,900 entries in AdGene that were supported by the Brain Initiative. So we're very serious about disseminating these resources very broadly. Next slide, please. And then finally, the Brain Behavior Quantification and Synchronization Program, which, as I mentioned, our goal is to develop and validate new tools and methods for analytical approaches to precisely quantify behavior and combine them with simultaneous recordings of neurophysiology. And we're using animal models, human models, sensor development, data coordination, and very, very importantly, we've set up a coordinating center to take care of the theory, computational models, and AI approaches that we'll need to kind of understand how all these data fit together. So next slide, please. But very importantly, while we're developing these foundational resources that will enable the field for decades to come, it's very important that we help people today. And we've seen an explosion over the last few years of brain-computer interface technologies and deep brain stimulation technologies that have been fueled by, I think, 3 related paths. One is the development of more precise ways of mapping circuits. The second one is the development of better recording and stimulation technologies. And the third is the development of AI technologies to be able to read out brain activity data to give us an indication of mood state. So one of the big challenges of treating mental illnesses and other more complex neuropsychiatric disorders is we have no biomarkers. But now we're getting to the point where through the recording of activities in specific regions of the brain, we now have activity biomarkers from this neural activity through AI techniques that can actually predict mood state, and the ability to now stimulate individuals suffering, for example, from depression, OCD, substance use disorder, into a better state. And we've also been able to, we've also seen some incredible advances in brain-computer interface technologies, for example, that allow patients who are paralyzed either through trauma or through ALS, for example, to be able to communicate with their loved ones and lead a more full life while they can. So just in the next slide is one stunning example of this, where a group in the BrainGate 2 consortium, so it's a consortium across the US. This study was based at the University of California, Davis. And what they were able to do is place four sets of electrodes over the motor and premotor cortex in a man in his 40s who was now becoming paralyzed from ALS, who was unable to communicate for his job with his loved ones. And what they've done is they have used the readout from this part of the brain as he's trying to enunciate words and phonemes and what have you. And through a very sophisticated artificial neural network, they're able to convert these signals into speech through some very, very impressive AI technologies, and then to have that drive a speech synthesizer. Now, we've seen over the last few years the development of similar, what we call brain-to-speech or brain-to-text technologies, but they require a lot of training and they're very error-prone. And the amazing thing about the study, next slide, is that after only 30 minutes of training, just about a month after the surgery, this gentleman was able to read out through this brain-computer interface 50 words with 100% accuracy. Unprecedented. And next slide. And then after just about another 1.5 hours of training, he was able to get through about 100,000 word vocabulary with about 90% accuracy. And with just a very little bit more training, that the accuracy went up to about 97%. So the importance of allowing people to communicate with their loved ones, for their jobs, just in general, is so important. But until we can have a technology that allows basically to be plug and play, getting this out to the thousands or millions of people who need it is going to be very difficult. This is one very important step in that journey. So I have a video, but I don't know, how's our sound? Yeah, good.
A lot of the demonstrations of VCI technology.
So the first video will show you his first training session that they had to stop because they were
The first
so
time we tried
astounded. Yep.
to use the system, it just worked.
Right.
Okay, ready?
That first time that he tried to speak and that word appeared on the screen, he started crying, I started crying, his family started crying, basically everyone. We had to pause, we stopped the rest of that block of experiment trials. Everyone composed themselves, and all right, let's do it again, and it kept working. So yeah, it was really special. It was a special moment.
People who do not have the ability to communicate well or quickly. are
This is the
often
gentleman
isolated and lonely.
speaking to us through
Having something
his
like this
brain
would give them
computer
hope and
interface.
opportunity to be more of themselves.
I think we're at a point where it does work well enough that it really lets him just be a part of society and to talk.
I am able to still work full-time for climate action, and I am able to do that well and more efficiently with this device.
Honestly, the reason this is so transformative is because it provides hope for people living with these kinds of diseases, that as they face their disease, that they know that they will hopefully have the ability to retain communication.
This was science fiction 10 years ago. So really quite astounding. We have a long way to go to bring these deep brain stimulation technologies for various disorders, brain-computer interface for people that are locked in and paralyzed. But this is a very important first step, and we're looking forward to finding ways of making this more accessible, more easily usable, for example, through, hopefully one day, through non-invasive technologies. But I think in the next 10 years, we will see an acceleration toward those goals, and we're very, very excited about that. This also brings up the issue of, as we are starting to probe the human brain, our own souls, the seat of our souls, we have to be very conscious and aware of the ethical implications of what we're doing. So the Brain Initiative takes this very seriously, this issue of what we call neuroethics, which basically describes the ethical, legal, and societal implications of what we do. Anytime we touch a human being, whether it's in the experimental realm or the clinical realm, of course, the primary concern is for safety. But as we start probing the human brain, we also need to start thinking about issues like agency, data privacy, about being able to read one day some aspect of human cognition. And this is something we bake in from the very beginning of the projects. We have program project teams that are actually focused on ethics specifically, as well as advisory panels that guide us in understanding and predicting the kinds of issues that we need to deal with up front before they become problems. The other very important aspect of what we're doing is to build a stronger workforce, and we take this very, very seriously. There's been quite a bit of very strong empirical evidence that teams that bring diverse perspectives, whether it's through lived experience or through professional expertise, scientific expertise, tend to outperform teams that have more homogeneous experiences. And we've now kind of incorporated this in what we do. Next slide, please. For actually demanding that our applicants tell us how diverse perspectives are going to make their science better. So this is something we take very, very seriously because there's so much talent around the world that is not being used to solve these very, very difficult problems. And they need to give us an action plan for how they're going to use diverse perspectives to do a better project so we can accelerate these discoveries and eventually toward cures. So next slide, please. And if you would like more information about what we do, well, you can ask Maurizio, which is the most important thing. So you can subscribe to our brain blog. We have a lot of great information on our website, but of course, the most important thing is to talk to the program officer. We love to interact with people to find ways that we can work with you. We do support international collaborations. It turns out it's a bit easier if you have the lead group who's applying be in the US, just for many, many administrative regions. But many of the projects I told you about here were conducted from labs across 3 continents and more. And we're looking forward to having more of that spread out because, again, we recognize there's talent that we need to solve these big problems from around the world. So finally, the last, next to last slide here, I invite you all to attend our workshop on the intersection of neuroscience and artificial intelligence. It's a hybrid workshop, so you can sign up and attend online. Or if you happen to be in the DC area at the time, we can try to get you in, but our seating is very limited. But nonetheless, this will be a hybrid meeting, and it'll be very exciting. I think we all agree that artificial intelligence is with us to stay, whether we like it or not. But the flip side of that is we can learn a lot about how natural intelligence works and apply that to developing better, more efficient, and more ethical AI algorithms for the future. So we're very much looking forward to pursuing this as an area for the years to come. And then finally, if you want more information about the BRAIN Initiative, just type in brain.gov and it'll take you right to our website. So again, it's been a tremendous honor to be here with you. I thank you so much for that honor, and I hope we can find ways of working together in the future. Yep.
After that wonderful, it's going to be a tough act to follow. But we're going to move on to the recognition for the best papers by a neurodiaspora member. So let's get started. First, to Dr. Diego Mendoza Halliday from University of Pittsburgh for his paper entitled A Ubiquitous Spectrolaminar Motif of Local Field Potential Power Across the Primate Cortex, published in Nature Neuroscience earlier this year, indicating the presence of a gradient in electrical activity across cortical layers preserved across brain areas and across human and non-human primate species. Dr. Mendoza.
There goes. Sorry.
Next up to Dr. Sandra Baez from Universidad de los Andes and the Global Brain Health Institute for her paper entitled Brain Clocks Capture Diversity and Disparities in Aging and Dementia Across Geographically Diverse Populations, published in Nature Medicine earlier this year, and discovering a signature of accelerated brain aging in large data sets of Latin American individuals ranging from healthy to frontopolar dementia Alzheimer's disease. And Dr. Baez is unable to join us today, but Dr. Reyes, who's also a co-author of the paper, will be receiving the recognition. And finally, to Dr. Bruno A. Benitez from Beth Israel Deaconess Medical Institute, or Medical Center, sorry, and Harvard Medical School, for his paper entitled Brain High Throughput Multiomics Data Reveals Molecular Heterogeneity in Alzheimer's Disease, published in PLOS Biology, and combining a multiomics approach with machine learning to leverage the heterogeneity of Alzheimer's disease. The authors extracted 4 distinct molecular profiles, with one of them corresponding to a significantly more aggressive cognitive compromise. I believe Dr. Benitez is here.
Okay, so for me, it's a great pleasure to give this recognition to Dr. Zulma Dueñas. She is a professor at National University of Colombia. She was a former president of the Colombian College of Neuroscience, who not only was my mentor when I was an undergrad student back in Bogota, but she also supported Neurodiaspora. So I think this is a very well-deserved recognition. Thank you very much. I'm happy that you're here.
take her because it's amazing for me.
Thank you. Moving on. I would like to now move to a recognition. Oh, thank you. Oh. Sorry, is that working? I don't know if I pushed the button. All right. So it's really hot in here. Okay. So as the Colombian College of Neuroscience and Neurodiaspora Group, it is our distinct honor now to remember and acknowledge the inspirational and paradigm-shifting career of Dr. Francisco Lopera. Dr. Lopera passed away this past September 10th at the age of 73, and his indelible impact received mentions across both scientific and general news outlets across the world. Born in the small town of Santa Rosa de Osos, Antioquia, in 1951, Dr. Lopera grew up in a region where memory loss, dementia, and other neurological disorders affected many families. In his own words, when you live in a community where people lose their memory and vanish into themselves, you cannot look away. You want to understand why, and more importantly, how to help. His proximity to those affected by the disease inspired him to pursue medicine at the University of Antioquia, specializing on neurology. By 1984, he had begun investigating cases of early onset Alzheimer's in local families. His research would eventually identify 25 families in the region who carried the gene responsible for the disease, leading him to become an internationally recognized figure in the field. He first encountered an extended family in Medellin who exhibited a striking pattern of early onset dementia. Before Dr. Lopera's efforts began, people referred to the condition as la bobera, the foolishness, and its occurrence was shrouded in superstition. The family comprises over 6,000 members, many of whom carry a presenilin-1 mutation. This mutation, which leads to the accumulation of amyloid plaques in the brain, causes Alzheimer's disease symptoms to appear as early as age 40, with some individuals developing the disease even younger. Lopera's meticulous work in documenting the clinical history of this family and tracing the genetic roots of the disease led to the identification of the PSEN1 E280A mutation, one of the most aggressive forms of familial Alzheimer's disease, and now a focal point of global Alzheimer's disease research. Dr. Lopera became a key figure in the Alzheimer's Prevention Initiative, API, clinical trial, a large-scale international research effort aimed at finding preventive treatments for Alzheimer's disease. The Colombian cohort of the API, launched in collaboration with researchers from the United States and Europe, was unique in its ability to study a population with a high risk of developing Alzheimer's disease at a young age. Among Lopera's most fascinating contributions was the discovery of individuals who carried the PSEN1 mutation but did not develop symptoms at the expected age. One case in particular garnered international attention, a woman in her 70s who had the mutation but showed only mild cognitive impairment despite the presence of extensive amyloid plaques in her brain. The woman volunteered to undergo comprehensive testing, including brain imaging and whole genome sequencing, to look for possible protective factors. This individual was found to carry an additional mutation in the ApoE3 Christchurch variant, which may have protected her from the full onset of Alzheimer's disease symptoms. The study was funded in part by NIH's National Institute on Aging, the Office of the Director, and National Institute on Neurological Disorders and Stroke. Results were published on November 25th, 2019 in Nature Medicine. This discovery could have profound implications for the development of treatments aimed at enhancing protective factors in at-risk populations. The findings suggest that therapies that reduce the binding of ApoE may be a potential way to treat and prevent Alzheimer's disease. In the words of NIH Director, Dr. Richard J. Hodes, sometimes close analysis of a single case can lead to discovery that could have broad implications for the field. Dr. Lopera's legacy extends beyond his scientific discoveries. First and foremost, he was a country doctor who started by doing house calls in the most remote places of Colombia and was known to the many families that participated in his studies as an approachable, humble, and warm person who leveled with them even when what he had to deliver were news that clinical trials had failed. Second, Dr. Lopera had a transformative impact on all the scientists and physicians that he mentored and trained. His Grupo de Neurociencias de Antioquia has risen to the top and is now known worldwide for its continued efforts in this research. Dozens of young neuroscientists have had their start in his group, but his influence has likely reached hundreds more. And some of them likely might find themselves in this room today. So please join me in keeping a moment of silence to remember Dr. Francisco Lopera. And the Colegio Colombiano de Neurociencias and the Neurodiaspora Group would like to present the Grupo de Neurociencias de Antioquia, a recognition for their significant contributions to the field of neuroscience. We honor the legacy of Dr. Francisco Lopera, and we celebrate the families that have contributed to the GNA's breakthroughs in Alzheimer's. Joining us from Colombia, we will make sure that you receive.
Thank you. Thank you very much for this recognition. I am deeply grateful with you. We recognize this work from Dr. Lopera. Dr. Lopera is a incredible scientific for Colombia and Latin America, and your discovery, all of her work is very important for the new discovery in protective genes and causality genes for Alzheimer's disease. Thank you, thank you, thank you for all my team. I am very grateful with you for this recognition.
You're up.
Okay. So actually, we changed to a more informal part of the evening. You probably have been waiting for this. I know I have. And then one of the goals of bringing all of these great neuroscientists together is, of course, just to know each other. And we don't know each other yet. So maybe, hopefully, you got one little paper at the entrance. It was either a neuron or a glia.
Yeah.
So I'm gonna kindly ask you to stand up, and perhaps we can make a neuron row here and a glia row right here, if you don't mind.
Wait, does everybody? Yeah.
Okay, thank you very much.
We might need to make some space so that the two rows can actually fit, probably from.
Okay, I'm sorry, I didn't know that I really want to
So you're gonna face each
because
other.
I
Neuron will face
enable
glia.
sitting with.
So we're gonna do a version of
Queríamos
speed
que al
dating.
principio fuera poder el himno y no estaba el doctor May todavía.
So all neurons,
I have seen on me.
are you? Okay, yeah, all neurons here and then all glia here, please. So please, make sure that you're facing someone. Okay.
Going to close the Zoom. Vamos a cerrar el Zoom. Muchísimas gracias a todos por estar.